Okay, everyone, good morning, and I'm really happy to be here, and I thank the organizers very much. We have to cut this off. To invite me to give this keynote in this session, and as you notice from my title, it's very broad. I'm sort of thinking about trying to incorporate everything, but obviously you gravitate to what you're most interested in such a presentation, which is the developmental aspect. It is really the ontogeny, where I will be focusing mostly on, specifically with regard to head morphology and flexibility of the phenotype in general. And in addition to my co-authors, I want to acknowledge long-term relationship with these people, and of course including a number of students that have contributed to my ideas and thoughts in this regard over the years, and their contribution is evident throughout this presentation. So needless to say, we are all fascinated by the diversity of Charrs, and me working on Arctic char, obviously, I have to emphasize that diversity, but we see it in all the different species, and this diversity is interesting, not the least because it is repeated local adaptations and local adaptive divergences in form of resource polymorphisms or even speciation. So, and interestingly, this polymorphism, and we've seen so many examples of that from all the different species in this conference, that is not necessary really to introduce it much further, but just that I want to conclude with saying that we are in the luxury of studying species which can represent better understanding of the evolution of biological diversity on Earth, which is quite a significant statement to be able to make. So obviously, we are very fascinated and occupied with the question of how does this diversity evolve? How is it generated? And we propose to use this framework of eco, evo, devo to approach the diversity. In this case, we are looking at Lake Charr as an example of diversity which is tracking the ecological environment down here in forms of local adaptations and plasticity. But let's then just very briefly look at how this works. We have abiotic and biotic factors in the ecological setting creating or being responsible for natural selection up here in the evo box. But this selection is feeding from phenotypic variation in the devo part. So we can picture this as a population of phenotypic diversity, which selection is able to feed on phenotypic variation, this arrow here. And then, of course, in the process of classical evolution, this is fed back in terms of genetic and epigenetic effects to the next generation of developing phenotypes. So I'm focusing on the developmental part here because that's sort of the subject of the presentation. But what is maybe most interesting, and for this particular presentation in particular, is the significance of this arrow here in terms of direct phenotypic plasticity from the ecological environment towards the developing phenotypes within generations. And again, often mediated through maternal effects. I'll get to the definitions of this later. Part of the maternal effects through egg size, for example, are being transmitted epigenetically as well, through what is called transgenerational plasticity. So these are the red things there are something that I will be focusing on for the remainder of the presentation, but I want to though emphasize that in recent years, we have had huge advances in understanding of how the developmental construction of the phenotype, sort of the evo-devo stuff, is now contributing constructively to phenotypic variation itself, including signals from genetic and environmental factors. And very briefly, just to connect this with this conceptual model of divergence, that for example, when we visualize the continuum of divergence within our Charr species or across the Charr species, we can locate them on various places along this continuum. But for the purpose of this presentation, focusing on plasticity and maternal effects, I am mostly focusing on the very early stages where we propose that these flexibility things are really at most working shortly following invasion. But then, of course, we propose also canalization down here, or loss of plasticity, and this will have relevance for some of the things that I want to talk about later on, sort of approaching my conclusion. But it cannot be emphasized well enough how the importance of the phenotype, because it is the phenotype that selection, natural selection and sexual selection is operating on. All this massive complexity of development is there included. And not only for fish, but very, very much for fish, and not the least Charr, we now know that a lot of the phenotypic variation characterizing juveniles and adults is actually constructed or determined during embryonic development or early juvenile development. So I'm now narrowing down the presentation sort of a little bit towards that part. And maybe the latest study I'm drawing our attention to is the study on Dolly Varden Charr from Lake Kronotsky in Kamchatka. So then let's just define these principal players in the presentation a little bit further. You're all familiar with this. Plasticity being the eco-devo arrow here is really the ability of the phenotype to respond to environmental signals like temperature, diet, very important environmental factors which of course are responsible for selection as well, and also are the important things that the phenotype needs to adapt to. So, and these signals are then interpreted by the developing phenotype in a complicated way, which I will not go into the details of here, but there are many different kinds of plasticity that can affect both the embryo and later stages, and all these things can then interact. And this has been very well documented for Charrs. There are some references here. The latest one being, the latest one here, Pepino et al. Being on Brook Charr. And this study is actually done in the field, and I mention that because that's I encourage such studies, because we need more field studies when it comes to studying phenotypic plasticity and early development. We have so many common garden experiments, so that's the point I really wanted to make about that. But now we got to maternal effects, which probably will play a greater role for the remainder of the presentation, maternal effects are defined as the influence of the maternal phenotype on the offspring phenotype, a very simple definition, both genetic and epigenetic effects. And they're also a source of what is called transgenerational, plasticity, which comes, of course, across generations here, where the environmental that influences the mother, for example, egg or yolk composition in eggs of the large eggs of salmonids, can be transmitted non-genetically across generations and affect the next generation, and even the second generation. But I highlight here yolk volume and composition of the yolk as influencing embryonic development, body size, behavior, and morphology of fish, which is basically where I'll be migrating now with my presentation. So we're talking about egg size and egg volume, and effects like that are very well known in Charrs. Here are some references. So just to then, very quickly, just remind you that these are the diverse environments. These are actually diverse environments in Iceland, where Charr are subjected to and what they have to adapt to, or environmental effects that produce plasticity and are responsible for even transgenerational plasticity in maternal effects. And then, yet again, introduce these fish, because now sort of introducing the fish I'll be talking about. You've seen this picture before in this conference, and different versions of it, with the 4 morphs of Arctic Charr in the lake Þingvallavatn in Iceland. Very dramatically separated morphs. And I draw your attention specifically to the morphologies, because that's what I'll be talking about mostly, and especially of the benthivore dwarf and large benthivore, which are benthic-derived morphologies, sort of, whereas the morphologies of piscivores, pointed snout, being more sort of probably closer to the ancestral phenotype. That's what we propose. So this just brief information has relevance on my interpretation of what I will say later. But of course, it is this environmental tracking of these morphs which is the main thrust of what's going on with the evolution of this diversity. And just to close in a little bit further, we are also studying, this is a very beautiful picture by Paul Vexi, as you notice, and it's a 10 centimeter approximately long small benthivore dwarf that is found all over Iceland, and Han actually spoke about their genetics the other day, and we have been studying those in all the many different local populations across Iceland in springs, where they are in all cases locally adapted. These are actually a cave form close to Lake Mivat. And then I draw your attention to these morphologies here of the piscivore Charr, adult piscivore Charr in Þingvallavatn and last benthivorm Charr. And the head morphology is what I draw your attention to. So both the juvenile sort of looking par marks and small size of the small benthivorm and this blunt head shape of the large benthivorm are pedomorphic characteristics. These are characteristics that are retained in the adult phenotype, like embryonic and juvenile characteristics which are retained in the adult phenotype. So here is some developmental proposal for you of pedomorphosis, or in fact it's heterochrony. So keep that in mind, you know, this especially the head shape, as like what is responsible for this in terms of evolution and development. But before moving into that, I want to show you these reaction norms from a fairly large common garden experiment on XP Charr progeny all across Iceland and in the lab exposing them to benthic and pelagic food. And the point I want to make is simply to illustrate that there is tremendous plasticity. So these morphs in these lakes, these are allopatric populations, have different reaction norms, suggesting also that this plasticity in diet-induced morphology, in this case, is adaptive and can evolve. But now we come to what I am going to focus most deeply on here, hopefully in interaction with all the others, because I am certainly in this presentation trying to also make you think integratively about these things. That's the underlying philosophy of what I'm trying to say. I may not succeed, but who knows? Is excise. And this is an illustration from an ongoing PhD of Samantha Beck, and where she is studying in details the consequences and significance of excises for the evolution of different morphs in Iceland. And some of these morphs have been mentioned several times during this presentation, for example, by Matthew Braham yesterday and Sigurður Snorrason. But the arrow, the blue arrow here, is really suggesting that we are here looking at morphs across which are becoming more specialized or more derived. And here are the excise variation of these morphs. This is actually an anadromous presumed ancestral population. So there is considerable excise variation. That is the main message, because I won't be talking much about Samantha's results here. She's building on some of the things that I'll be talking about in this presentation. We'll see her work later. But her main sort of underlying hypothesis in her work is that with more derived morphs, you have narrower distribution of excise. So that was to convince you that excise is very likely a significant player in the evolution of different morphs of Arctic Charr. So then let's go to and look at what we classically know, and all of us are aware of, that first of all, salmonids and chars have large eggs, from 3 to 6 millimeters. These are huge eggs compared to many, many other fish species. So they have great consequences for size of embryos. So here you can see an illustration. You can actually see the size differences here. This is from Camille Leblanc's PhD study. And at first feeding, the small eggs produce about maybe 2 to 2.2 centimeter long fish at first feeding, whereas these are maybe 2.5. This is just to give you some visual information about what's going on. And she finds that there is a very close correlation between size of embryos at hatching and size of embryos or juveniles at first feeding. And we know from her studies and other studies that these can have dramatic life history consequences for later growth, maturation patterns, and so forth. So this is a very important maternal effect, to say the least. But one would expect, of course, and that's pretty obvious, and classical studies have shown that excise and embryonic development are closely related. And this builds to a great extent on Waddington's work, his epigenetic landscape idea that embryos with little yolk must prioritize development of essential features, such as respiratory, gut, and trophic structures. So this basically is already suggesting that we are, in the embryo, is dealing with a resource which varies in its limitations. So if there are limited resources, the embryo has to prioritize development of certain structures which are immediately suggesting that they may be less flexible. So I'm giving you a hint to my conclusion or my interpretation of all of this. Thus, we can expect that there is a relationship between egg size and embryonic development in an Arctic char, or in char in general, or salmonis for that matter. And we did a fairly detailed study a long time ago with Arctic char embryos, and we found out that embryos in small eggs actually developed relatively faster. In this case, they were producing melanophores on the head and melanophores in the eyes that were measured. There also the heart started to beat earlier in smaller embryos. So this prioritization hypothesis or fact was really being demonstrated there. So we would, of course, expect that these effects of timing, or heterochronic, we could say, of ossification of skeletal elements, for example, in the skull of Charr, which is so morphologically variable, would be affected by egg size. So let me now convince you that there is that ossification of the skull and the bones that is significant when it comes to the sort of development of morphological differences of the skull in juveniles and adults. First of all, a very recent study by Dahl from Dollyvarden Charr, again, where they saw a very clear association between timing of ossification of skull bones and later juvenile and adult morphologies. So I urge you to look at that evidence. Now this, of course, was not association of egg size, was not there. I'm just demonstrating that this timing is actually a fact. But a much more detailed study by Kevin Parsons on Malawi cichlids showed that embryonic skull ossification was accelerated by elevated wind signaling, a wind pathway which influences timing of ossification in these fish. This was actually at embryonic stages, but it produced a novel phenotype, which was a really, really very blunt snout, almost deformed adult fish. So here we see the early larva with very blunt snout, very highly ossified when there is high wind signaling, and much less ossified and not nearly as blunt when there is low wind signaling. So I'm just using this to convince you that timing of skull ossification is very, very significant for later morphologies. And not only that, they concluded that this accelerated ossification locks into place an early larval phenotype, which suggests that could be the mechanism responsible for creating those different adult phenotypes. So there's locking in. So here we then connect this with actually an older study done by Guðni Magnús Eiríksson, comparing the ossification processes that we knew at that time were actually somewhat different between the Charr morphs in Þingvallavatn, between crosses of the small benthivore and the planktivore and their hybrids. So here are the crosses, A, B, C, and D. And what he looked at was the timing of ossification in embryos at different stages, shortly after hatching until about first feeding. So this is just to show you here, this picture, that he stained the embryos for cartilage, the blue, and bones with alizarin red. So the red color is then being scored here for stage 1, low, and stage 4 with high. And because there's lots of information in this slide, I'm just drawing your attention to one thing here really. At first feeding, we see that ossification, the pure benthic progeny with the blunt snout, is much greater than in the pure planktivore progeny up here, the dark. But then something else is going on in the hybrids. Certainly there's an evidence here of a genetic component of timing of ossification, but the fact that planktivore mothers, when you have planktivore mothers and small benthivore fathers, there is actually less ossification going on, and much greater ossification when you have small benthivore mothers and planktivore fathers. Now I'll tell you that the planktivores have larger eggs, considerably larger eggs than the small benthivore, so this is clearly a maternal effect, affecting the timing of ossification in the bones of these Charr. So we can conclude from this that it's highly likely that the pedomorphic head morphology of some of the benthic Charr morphs, and these are morphs you see not just in Arctic Charr, we see them in Lake Charr and Dollyvarden Charr in particular, is could be determined by earlier skull ossification, locking in the phenotype, to sort of use Kevin Parsons' interpretation. So, which means that we would propose that mores like large benthivore char are more canalized, with, that is to say, less plastic than the more pelagic phenotypes. And that this could be partly influenced by different yolk volume and egg size. So that's sort of the conclusion here. But then let's look at the other side of the story. And what about the flexibility then? Here we have provided some evidence that timing of bone ossification in the skulls of these char could be leading to some narrowing of the flexibility and presumably less plasticity in morphology and presumably other things in the phenotype. But we know from many studies that direct embryonic development, usually involving larger embryos or juveniles, is generally associated with larger eggs. And there are classical studies of references that indicate here. But Wake and Hanken did a detailed study on the evolution of direct development, including larger eggs in salamanders, and found out that this involved great release of developmental constraints. Sort of, if you think back to Waddington's epigenetic landscape thing, that when you have more resources to work with, there are more opportunities to develop. And not only develop direct development, but also greater variety of structures can be created in that type of phenotype. And we saw some evidence of this in Guðni Magnús Eiríksson's study that I showed you earlier, that the planktivore progeny coming from larger eggs than small benthivore progeny, that was the other half of his study, they were clearly directing their energy much more to embryonic growth than to skull ossification. So the egg size seemed to make them be able to use this trade-off. They were not sort of constrained by having to direct their energy towards the trophic structures. So what we can then propose, it's a very simple prediction or hypothesis, that is that larger embryos can in fact be more phenotypically plastic than smaller ones. And bingo, when I was writing this presentation, I found this paper and on Brook Charr. It's a very detailed study done by Penny et al. On individually raised embryos of these fish in the laboratory. And they found out, and they raised them at different temperatures and different salinities, and found out that larger embryos from larger eggs were more plastic than smaller ones, exactly as we would have predicted. But what was important here, and for the whole interpretation of this kind of finding for early development and reproduction, is that these maternal effects are interacting with abiotic factors such as temperature, which are known to affect embryonic development in fish. So here is a huge fitness-related phenomena. We can just start to think about spawning in char and how that is seasonally constrained, and temperature is a very, very big player here. So we propose that we really need to take the local incubation environment and its association with egg size in consideration when considering embryonic development, especially in the field. And that's sort of the reproductive part. So we won't be able to get any further with that here, but that I would like to really work that out further here with you. And that is one of the reasons why I'm emphasizing that we should be doing more experiments, more experiments in field conditions. I hope you understand what I'm trying to say. But I will now conclude, because these results from Penny et al. Really elevated the significance of our earlier findings, in this case a PhD study by Camille Leblanc, again, my colleague, where she totally removed the genetic factor and worked with families or intra-egg size variation on a family basis, and in this case, actually on an aquaculture strain. So let's just look at her findings there, because they are having some significance with relation to what I've already told you. She compared 4 different families, here, here, and here, sorted them in large, the blue, and small eggs, and then looked at their morphology, actually shortly after they started to feed, when the fish were about 4 or 5 centimeters long. And what she found that there was a consistent difference in morphology in the progeny, where small embryos or from smaller eggs, juveniles from smaller eggs, had slightly more benthic morphology actually than progeny from larger eggs. So it's kind of perhaps suggesting the idea that this locking in of the phenotype is happening because of different egg size. But what would be more interesting even is her results on the behavior of large and small embryos at first feeding. And actually this remained over juvenile period, where embryos from larger eggs or juveniles from larger eggs showed much greater behavioral flexibility in an experiment feeding from the bottom and surface foraging. So obviously, where the embryos from the small eggs were much less flexible, much less plastic presumably, and ate much more from the bottom. Of course, that could possibly be related to their morphology as well. Now I'm just speculating. But the main thing is that there is a huge difference here in their feeding behavior, and you can just imagine what consequences this can then have for, let's say, a situation during early divergence following an invasion of an ecological opportunity of Charr. To have this type of mechanism at your hand. And just very briefly to tell you that a lot of the work being done in our groups is focusing on gene expression and genetic differences, as you already learned, for example, from Skúlason's and Hans' presentation. And we know that there are many genes being expressed in the head of embryos, genes that are related to ossification processes. And when it comes to egg size, Samantha Beck has actually already found one regulatory gene being expressed related to egg size, which is responsible for osteogenesis. And this has been well studied in cichlids from Lake Tanganyika as well. So maternal effects are clearly interacting with the developmental program of the embryo, and interacting with plasticity and interacting with the genetic material as well. So that is the integrative statement for you. But to conclude then, we can say that plasticity and maternal effects can strongly influence Charr salmonid phenotypes. I want you to understand that I am not trying to explain everything by this. I'm just drawing your attention specifically to the significance of these factors. You know that large eggs produce larger progeny that can be more flexible than smaller ones. Of course, suggesting then that the smaller ones will be less plastic and possibly even creating tendency towards pedomorphic shape in the benthic Charr. And that this can obviously contribute to resource polymorphism and speciation, and maybe needs to be taken more into account. So, and finally, I mentioned this philosophy of integration, the eco-evo-devo. We certainly want to promote that even further, but just to conclude then with this and locate our sort of the story that I've been trying to tell here in this presentation to this diagram here. Well, we, I think, have evidence now to really support the idea that egg size and plasticity are playing interactively a very important role here, presumably with behavior being very, very important, affecting also development of skull processes, just to look at the Devo part, leading to more canalized phenotypes. And I didn't say it very clearly on a specific slide before, but Kevin Parsons actually did a detailed plasticity experiment on the benthic and the pelagic morphs in Þingvallavatn, and he found that the benthic morphs were more derived, were more canalized, sort of down this road here. And of course then the excitement is to follow this all up with connections with the ecological environment in each case, and for the developing embryo that would be the incubation environment in the field. So, thank you very much.
So I think we could take a few questions, if anybody has any.
So would it be reasonable then, based on your findings with the influence of egg size, to project it in a situation where, for example, with brook char facing higher metabolic costs and due to warmer temperatures, that if that resulted in females producing smaller eggs, that it would have an additional effect of reducing plasticity going forward as a consequence of climate change.
I would completely agree with you, and I have really nothing to add to what you said. I think this is exactly the point, that we need to take these exercise dynamics into account when considering climate change. We know now, I could go on and on explaining some other things, but we now, for example, have evidence that we have one morph spawning in Iceland at a relatively high temperature site, and these guys have huge eggs.
Skúli, I just wondered whether you had published or were going to publish that synthetic diagram that you ended with.
Yes, we actually, there is a review and sort of idea paper in being reviewed now by a journal, and we hope to succeed in having this published in a few months.
Good.
Skúli, I have a question. So you talked about egg size, but have you guys looked into egg quality? Because there's quite a body of literature on trade-offs between size and quality of the eggs.
Yep. Yeah, definitely, and that's something I had to leave out here. But we have done both sort of energetic measurements of egg sizes in these morphs, so that's what Samantha is including in her work. So if you just look at it in terms of egg densities, apart from all the hormones and whatever may be or enzymes that are operating there, you now, for example, know that the large benthivorte Charr of Þingvallavatn, just to give you an example, they do have a denser yolk. And mind you, these fish are spawning in an environment which is completely constant temperature. And they need to have a different mechanism potentially of locating, or let's say timing their first feeding and developmental events. So, I mean, we are obviously looking at very complex signaling mechanisms in the egg or buffering mechanisms of that sort. It's not just size, as you said. But the size, of course, is important for the mechanical processes, because we also have to think about the distances that cells have to migrate and things like that. So huge distances considering the variation in these eggs.
All right, well, thank you, Skúli, very much.
Yeah, thank you.